Good afternoon, everyone. My name is Aihong Yang. I'm from Tianjin University of Traditional Chinese Medicine, China. I'm honored to introduce my studies to you. The title of my presentation is A Multifunctional Anti-AD Approach, Design, Synthesis, X-ray crystal structure, biological evaluation, and the molecular docking of chrysin derivatives. Firstly, let me introduce my co-authors. Chang Liu is my graduate student. Rui and Xiaodi are my co-workers. Our group has published 11 papers about research of anti-AD natural products in recent 3 years. Chang is an excellent student. She is specialized in crystal culture of natural products and further structural analysis. Recently, we jointly published a paper about chrysin derivatives in European Journal of Medicinal Chemistry and this paper. Next, I will start my report. Alzheimer's disease has many manifestations and pathologic features, such as memory loss, disability, dementia, depression, etc. So, with the aging of the population intensifying, Alzheimer's disease has many manifestations and pathologic features, such as memory loss, disability, dementia, depression, etc. So within the aging of the population intensifying, finding a cure or reasonable treatment for AD has become an urgent priority. Multi-factors have been implicated in the etiology of AD. They might contain acetylcholine deficiency, biometals, dyshomeostasis, oxidative stress, beta amyloid aggregation, neuroinflammation. Although the exact pathogenic mechanism of AD is still unknown, numerous research efforts have increased the understanding of the disease at the biochemical, cytopathological, and genetic levels. Multiple pathological factors are believed to be interconnected and tightly associated with metal ions like iron, copper, zinc. Many reports emphasize the roles of metal ions in the pathogenesis of AD. Metal ions are essential for life, playing important roles in the human body. In nature, nearly half of all proteins, metal binding proteins called metalloproteins. Metal dyshomeostasis directly causes neuronal dysfunction, leading to neuronal cell death. Clinical studies have shown overloaded levels of copper, iron, and zinc ions in post-mortem brain tissues of AD patients. Metal ions are also highly involved in other pathogenic pathways in AD. For example, metal ions can induce amyloidogenic pathways, which means APP is cleaved with beta or gamma secretase and generate neurotoxic A-beta peptide. Once the A-beta peptide is generated and secreted into the extracellular space, it spontaneously transforms into fibrils called amyloid plaques and A-beta aggregation. Metal-induced A-beta aggregation could result in neuronal cell death, synaptic dysfunction, and cognitive impairment. The generation of ROS is also strongly related with redox-active metal ions, especially copper and iron ions. Redox cycling between the redox couples can facilitate the activation of molecular oxygen, which is largely mediated by A-beta. Therefore, more effort is still demanded to explore new strategies based on metal-related hypotheses. Metal-related therapeutic strategies have attracted more and more attention for AD therapy. Some metal chelators capable of targeting metal-induced A-beta aggregates and ROS generation, while chelating A-beta bound metal ions have been developed. Metal complexes capable of occupying the metal binding sites of A-beta have also exhibited promising potential for AD therapy through affecting the A-beta aggregation and the ROS activities. And there are many nitro metal chelators in TCM which have aroused our research interest. After more than 2,000 years of development, TCM has accumulated a lot of practical experience in senile dementia, that is AD in Chinese medicine. The most commonly used Chinese medicine for the treatment of AD are, the Chinese name is 人参、远志、蜀地黄、甘草、杜仲、丹参、益智仁、白芷. These herbs contain flavonoids, xanthan, anthraquinone, coumarin, eosinic acid, and other compounds. These compounds have the common features that they all have metal chelating sites that can coordinate with metals. Therefore, these compounds can be used to treat AD by regulating metal homeostasis. In recent years, our research group mainly focuses on the screening of natural metal chelators for the active components in TCM. Here, the coordination chemistry of TCM is introduced first. The theory of coordination chemistry of TCM means that the effective chemical components of TCM are neither pure organic molecules nor pure metal ions, but mainly coordination compounds composed of organic molecules and metal ions. Compared with organic molecules and metal ions, the complex is more representative of the overall effective components of TCM. It can be comprehensively represent the active center of TCM. The theory of coordination chemistry of TCM is based on the fact that the structure of the active components of TCM mostly contain nitro chelating groups such as hydroxyl and carbonyl groups, which can coordinate with metal elements in the organism to produce special pharmacodynamic functions. For example, our group has done research on chrysin, naridinium, amidine, coumarin, eosinic acid. As you can see in the first 4 compounds, they all have carbonyl group and hydroxyl in their position, which can form a chelate ring with metal ions. And eosinic acid has 2 carbonyl groups as chelating sites. Metal ions in living bodies mostly exist in metalloproteins in the form of the complex and maintain close coordination relationship with organic molecules or biological macromolecules. Because many AD-associated targets are capable of such coordination with metals, natural chelators and their metal complexes open up a new pathway for the treatment of AD. The structural characteristics of nitro products are summarized as follows. Regulating metal homeostasis, it has heteroatoms such as nitrogen, oxygen, sulfur, etc. And there are 2 or 3 connecting atoms between heteroatoms, which can form a stable chelating ring with the metal center. This aggregation of A-beta, it has planar conjugate system, which can form pi-pi stacking or pi-cation stacking with A-beta, and then insert into the beta fold of A-beta molecule. Antioxidation, it has phenolic hydroxyl groups that form stable radical structures or groups that readily give electrons. According to the multi-target treatment strategy, our research group modified the structures of the screened nitro ingredients to increase their biological activities. Tertiary amines amine group as a kind of classical bioactive group can significantly improve CHE inhibitory activity and antioxidant activity. Firstly, for chrysin, the 7 hydroxyl group was replaced by amine groups to obtain a series of derivatives. And when the 5 and 7 hydroxyl groups were replaced by carbamate group, another series derivatives were obtained. For nitrogen, one or two hydroxyl groups were replaced by amine or carbamate groups. 9 nitrogen derivatives obtained. In the same way, we modified the structures of eosinic acid, xanthone, coumarin, and other components to obtain corresponding derivatives. And we carried out multi-target anti-AD activities tests, and the activity increased after modification. Here, we take the active ingredient of Yizhiren, a traditional Chinese medicine for the treatment of AD, as an example to explain the results of anti-AD in detail. Yizhiren, one of China's 4 southern medicines, has been regarded as a classic herb for treating dementia since ancient times. Its active ingredient, chrysin, is one of the important nitroflavonoids in the herb, and it's also found in many other plant and bee products. And it has the characteristics of abundant source, easy to obtain, and high feasibility of research. Recent studies have shown that chrysin has a variety of biological activities, including anti-inflammatory, antioxidant, and neuroprotective effects. Chrysin has a planar structure that can effectively insert between A-beta proteins and inhibit their aggregation. There are carbonyl and hydroxyl groups in chrysin, which can be used as natural metal ion chelating sites to effectively chelate metal ions and maintain metal ion homeostasis in the brain. And this figure shows the process of our research. Through nucleophilic substitution, alkyl amine groups were introduced into the 7-hydroxyl position of chrysin, and a series of derivatives were obtained. And then these derivatives act as ligands and react with metal ions to obtain metal complexes. Notably, the single crystals of compound 1 copper and 5 copper were obtained and analyzed by X-ray crystal diffraction. Next are the results from the metal chelating studies. The selectivity of metal chelating is extremely important in the choice of the suitable chelating agent. The chelating ability of the chrysin derivatives with copper, iron, zinc, aluminum, magnesium ions, etc. were evaluated by the UV visible spectroscopy assay. The red line is the spectrum with addition of the copper ions to the ligand, and the green means iron ions, purple zinc ions, orange aluminum ions. When these ions were added, the spectra changed significantly, and the peaks red-shifted. The results show that the ligands could selectively chelate with copper, iron, zinc, and aluminum ions. The possible process of in vivo conversion of copper ions may be the unbounded chelator coordinate to the metal ions to form stable complex. It is then metabolized out of the body. The stability of the stability constant of complex 1 copper was calculated to be 1.715 times to 10 to 10. It is speculated that in AD brain, the ligand may remove copper ion in overload situation. Ligand 5 is a chrysin derivative formed by introducing a morpholin into 7-hydroxyl group. It is a novel compound, and we got the single crystal of it, and the asterisk structure was shown here. And here is the stacking diagram of it. You can see the pi interactions formed between the molecules. When 5 coordinated to copper ions, the complex formed. Notably, we obtained the single crystal of the complex and analyzed the structure. The complex consists of 2 ligands with 2 copper ions and 6 chloride ions. Each copper ion is coordinated with a carbonyl oxygen and 4 chloride ions to form a pentagonal geometry. The 2 copper ions are branched by chloride ions to form binuclear copper complex. 2 ligands in a molecule lie parallel to each other in 2 places. The 2 or 3-dimensional structures are formed by peptide interactions. Furthermore, Ligand 1 is a chrysin derivative formed by introducing a dimethyl amino into 7-hydroxy group. We also obtained the single crystal of the complex of 1 and copper. The novel 1-copper complex was characterized by single crystal X-ray diffraction. The figure showed that the 1-copper complex was crystallized in the monoclinic system. The molecular structure exhibited 1-copper center, coordinated by 2 ligands in trans fashion to generate an almost perfect square planar geometry. The copper center was full coordinated by 4 oxygen atoms to form a square. 2 ordinated items are from the phenolic hydroxyl and the other 2 from the carbonyl group. The chelate ring formed by the ligand and the metal was almost symmetrical. This structure indicated that 1-copper was an inner orbital complex with dsp2 hybridization, which further indicated that the oxygen atom as coordination items had a strong electron donating ability. The molecular planes in the stacking structure were not arranged in parallel, but form an angle of about 50 degrees. So there were no pi-pi interactions. These layer structures form a 3-dimensional coordination network through various non-covalent interactions. The analysis of the single crystal of the complexes show that the chrysin derivatives could chelate metal ions, and it is very useful to the analysis of the structure-activity relationship. For example, 1-copper complex formed a spatial layer structure that can be inserted between A-beta proteins to inhibit the aggregation of them. Then, the multi-target anti-AD activity of the derivatives were studied. As shown in the table, chrysin as the parent compound did not show obvious anti-CHE activities, while the derivatives obtained remarkably improved inhibitory activity. The structure-activity relationship clearly indicated that the tertiary amine moieties were the key pharmacophore to inhibit both ACHE and BOCHE. They were selective inhibitors toward BOCHE. Compound 1 as the most promising candidate exhibited the highest selective BOCHE inhibition. To gain further insight on the mechanism of inhibition of this class of compounds on CHE, kinetic studies were performed on the inhibitor 1. The results in the right figure indicated that compound 1 was least type inhibitor of competitive inhibition and non-competitive inhibition that could simultaneously bind to CS and PAS of enzymes. Next, several assays were employed to determine the antioxidant capacities of the compounds. The left table shows that all compounds have good hydroxyl radicals scavenge activity. DPPH scavenge ability of the copper complexes have been improved compared to the parent compounds. As shown in the right table, the text compounds exhibited moderate ability to reduce peroxyl radicals. Cyclic voltammetry is a commonly used electrochemical research method. It can be used to study the properties of electrode reactions and the mechanism of antioxidants. The electrochemical properties of these compounds exhibited a closed loop, which indicated quasi-reversible redox processes, and it is proved that the compounds have potent superoxide radical scavenging activity, and the activity is in concentration-dependent manner. As you can see, with the increase of the concentration of the sample, the peak from the current lower down, and the mechanism of these compounds may be single electron transfer. In addition, the inhibition activities of A-beta aggregation were measured. It was mentioning that the inhibitory activity of these compounds on copper-induced A-beta aggregation were around 80%. And the inhibitory activities of other kinds of aggregation were only around 40 to 50%. So the aggregation kinetics were studied to further explore the mechanism of action. Figure D shows that one is able to inhibit the total amyloid species formation in a dose-dependent manner. To examine the potential cytotoxicity of compound 1, the PC12 cells were exposed to 1 at different concentrations. As shown in this figure, the cell viability of 1 was 83% at concentration of 10 micromolarity after 24 hours of treatment. Therefore, it is safe and reasonable to conduct a biological experiment in the range of this concentration. Molecular docking experiment of the compounds were carried out on ACHE, BUCHE, and A-beta. The results show that the derivatives had a certain affinity for these biological targets, and the type of interaction between small molecule inhibitors and amino acids were further defined. Also, these results predicted in silicon indicated that these compounds have suitable drug-like characteristics, brain availability, and plural bioavailability. Taken together, the multifunctional properties of chrysin derivatives may provide a useful template, and one might be a promising advanced lead candidate for the development of new anti-AD drug. Furthermore, we also synthesized another series of chrysin derivatives based on the hidden multi-target strategy. The purpose of synthesis is to protect phenolic hydroxyl group, because metabolic studies have shown that the active phenolic hydroxyl group may be oxidized in liver by means of glucuronidation, sulfation, and methylation, leading to a decline in bioavailability. Therefore, we adopted a pro-drug design strategy to protect the phenolic hydroxyl group of chrysin, to enhance its lipid solubility and the permeability of BBB. The derivatives were obtained by combination with chrysin's metal-chelating site and reverse Tigmin's anti-CHE pharmacophore to form an ester, which improves the hydrophobicity and protects the phenolic hydroxy group at the same time. 4 derivatives were synthesized as the hidden multifunctional agents for AD therapy. These derivatives have the similar ester structure as reverse Tigmin, which can be hydrolyzed by CHE to release the hidden active molecule, chrysin. They were fully characterized by proton NMR, carbon NMR, MS, and elemental analysis. Then the anti-AD activities of the chrysin derivatives were evaluated. Firstly, the CHE inhibitors activities and the kinetic analysis were studied. Most compounds showed moderate to good inhibitory activities to CHE, especially to BOCHE. Compound 8 was demonstrated as the highest selective BOCHE inhibitor. The sequence of inhibitory effects of these derivatives on BOCHE was as follows: 8, 7, 6, 9. Based on the structure-activity relationships studies, it is clear that electronic properties of tertiary amine substituents play important roles in the inhibitory activities. These results may be closely related to the pKa value of the tertiary amine moiety. The kinetic studies of the mechanism of compound 8 show that it could simultaneously bind to CAS and PAS of these 2 enzymes, indicating that it was promising compound as a double binding site inhibitor. To verify whether derivatives could be hydrolyzed by CHE in conditions similar to cerebrospinal fluid, we carried out hydrolysis experiment of BOCHE by Hplc using compound 8 as an example. As shown in the figure, when H was incubated with BOCHE at 37 Celsius for 48 hours, small protein of chrysin could be identified by Hplc. It showed that it could be hydrolyzed to chrysin in the presence of BOCHE. The chelating mitoability of the hydrolyzed product chrysin was studied by UV. Chrysin could chelate copper, iron, zinc, ions. These results indicate that it may have the characteristics of drug. Next, the effect on the free radical scavenging properties were studied. From the table, you can see all compounds have potent hydroxyl radical scavenging activity. The excellent anti-activity may be related to the formation of conjugated system between the 2 and 3 position double bond of the C ring and the 4 position carbonyl group. It is helpful for the formation of more stable free radicals, thereby interrupting the chain reaction. And then cyclic altimetry was used to test the scavenging ability to superoxide radical. As shown in the figure, the reduction oxidation current formed a closed loop. It indicated that the reaction process is reversible. The height of each peak is proportional to the amount of the superoxide radical. The CV was measured after the addition of antioxidants and the magnitude of the decrease in the oxidation peak current from the blank peak indicated the amount of the superoxide radical decreased. The inhibition of A-beta aggregation was studied, and curcumin is a reference compound. As shown in the left figure, all the tested compounds showed a certain inhibitory effect on the aggregation of A-beta. Compounds 6, 7, 9, chrysin had a slightly stronger scavenging effect than curcumin. The scavenging effect of 8 was weak. The inhibition of copper-induced A-beta aggregation by the compounds is shown in the right figure. The compounds have strong inhibition on copper-induced A-beta aggregation, and the inhibition rate reached nearly 80%. And then in order to further explore the possible inhibition mechanism of the most promising compound 8 for ACHE, BOCHE, and A-beta, molecular docking simulations were carried out. The results of molecular docking also speculated that the compounds had a certain inhibitory effect on ACHE, BOCHE, and A-beta. In addition, it is necessary to predict the AD, MET, BBB, and the oral availability. The results showed that the most compounds exhibited drug-like characteristics according to the Lipinski's rule of 5. AD-MET prediction results indicated that most compounds have appropriate pharmacokinetic characteristic to penetrate BBB, and 8 was predicted had a suitable physical chemical for oral bioavailability. In summary, amine and carbamate derivatives of chrysin were synthesized based on multi-target drug strategy. And most of them show the CHE inhibitory activity, strong antioxidant activity, and A-beta aggregation inhibitory activity. Some compounds show the potent selective inhibition of BOCHE, which may play a therapeutic role in treating the high BOCHE activity in the brain of patients with advanced AD. X-ray single crystal analysis of metal complex provides reliable basis for structural activity relationship analysis at a molecular level. We hope these results could provide new ideas and methods for the research of innovative TCM drugs based on natural products, and also provide certain molecular models and theoretical basis for the design and development of multi-targets anti-AD drugs. Finally, thanks very much for your listening. It is a great honor to communicate with all of you, and I wish the conference a great success. Thank you.